materials that have been developed for nanoparticle delivery interact with cells, and often many of us use cell lines to do our initial studies. And then we're moving more and more towards 3D spheroid systems, and I'll be talking about the positives and negatives of some of these things, as well as then animal models, with the ultimate aim to get to humans. But the complexity from here to get to here is enormous, and you can imagine that you're not just thinking when you do even biodistribution studies or efficacy studies in a mouse. There's still many challenges to get it into a human. So we try and maximise our ability to develop these materials and test them here, so we can ultimately get to here. Now, as many of you know, that there's many challenges to cancer delivery. I guess controlling the size of particles, preventing aggregation in vivo is very important, because if you don't, you can have severe liver and kidney toxicity. The biocompatibility, how long these materials are actually circulating, again, is very critical. And it's also important to, I guess, understand the immune system in this process, because you want enough time and enough material to reach the disease site and release its cargo in order to have efficacy. But you also want to be careful not to overstimulate the immune system as well, and give a major problem. But it is generally believed a small increase in activation of the immune system may not necessarily be a bad thing, as long as it's not severe. Other practical challenges, I guess, to think about for clinical translation is really, it comes back down to the disease state, and really, I guess, understanding the disease and the biology of disease you're working on. In the case of cancer, you want to try and deliver things directly to tumours. You want to make sure that the cells can take up the particles, they're localised to the right site, you're not getting it distributed to areas of the body that you don't want it to be distributed to, and you want controlled release and functionality of these materials. I mean, one of the big challenges in cancer therapy that many of you would understand is that current chemotherapies are highly toxic. So even though there's been advances in immunotherapy and some targeted therapies, these are not without side effects, and particularly when it comes to chemotherapy, it becomes a major problem. So in the children's hospital that I'm associated with, in the children's cancer ward, at any point in time, 50% of the kids in that ward are in there because being treated for side effects of chemotherapy, not being treated for their cancer. So it tells you something about the clear need to develop more effective, less toxic therapies. So that's why it's important when we're developing these materials that we always constantly keep in mind to reduce the toxic side effects of the treatments. I just put this in here to really say that there's a large variety of nanomaterials and different delivery agents and sensors that are used, and each of these will have its own advantages and challenges as well. And again, picking the right system to actually investigate how your materials might be working and how effective they will be is quite important. So for the chemists in the audience, you know that the really recognising and characterising the physicochemical properties of the nanoparticles is important. But I think more and more we're starting to understand how these properties are starting to affect the tissue particle type interactions. Including things like shape, surface charge, size. Shape's twice. I must have thought it was important. And drug loading and release. So these are important, I guess, characterizations we normally do before we actually go into any biological system. Many of us use cell models to do our in vitro testing, I guess, to look at the binding, the uptake, the release, and even the efficacy of the materials as well. And in part of my talk, we'll be talking about those models and what we need to think about. The other thing is the toxicity of these materials, and often people will just do a quick cell viability assay, and sometimes they may look at cell growth, or I think not as often, and some basic inflammation studies, and also possible genotoxicity as well. But again, if you look at a lot of publications, when they've got a new blockbuster nanoparticle, they don't cover all these type of things, and it's actually quite important. It's important to also work with pathologists when you go into your animal models to make sure these effects are not happening in vivo. Oops. Thank you. And then the other issue is the in vivo properties of these materials, the biodistribution, the toxicity of them, the pharmacokinetics, the efficacy. You heard earlier about Doxil. I mean, one of the key features there is that the change in the biodistribution of what is a highly effective drug. But one of the big problems with that drug was that it's associated with cardiotoxicity. I mean, it has other toxic side effects, but that's probably the number one. With many people getting cardiac damage as part of the treatment. So if you can change the biodistribution of some of these agents, that's even better. But if we can better target them, and the other, I put all these different animal models down because again, it comes down to what you're investigating and what you're studying as to which models you're likely to use. I mean, the most common one we use in our laboratory is the mouse model. So in vitro cell models, so I guess the majority of people, and for many decades, we've been using two-dimensional cell cultures where tumour cells are grown on a plastic surface with media, and we've studied multiple properties in those cells. It's really valuable when you've got these models to look at nanoparticle uptake. You can see whether things are getting trapped at the lysosomes or the endosomes. And what's going on. So they're convenient for that purpose. But when really trying to understand, I guess, the biology of what's going to happen in a real-life tumour model, they do have a lot of limitations. So they should just be used as a stepping stone to do more complex models. So we know that tumours grow in a very complex environment, and this is an incredibly simple diagram to depict that. Normal vasculature has got these intact blood vessels. You've heard about the EPR effect. While in tumours, many times these blood vessels are disordered and leaky. But one of the key features about tumours is that they often have a necrotic core, depending on the size of the tumour. So near the blood vessel, there's a lot of oxygen here, but as you go towards the core of the tumour, they're deprived of nutrients and they're deprived of oxygen. So it's a very hypoxic environment. Stem cells have a great ability, quiescent cells have an amazing ability to survive there, and drug-resistant cells can also survive there. But in this complex environment, you also have immune cells, you've got stromal cells, you've got other interconnecting cells in that system as well. So again, very complex compared to what we often analyse when we're trying to study these materials. So it's always good just to keep this in mind and understand the limitations of what's being done. But one of the advantages, I guess, of this is that we can partially start to mimic this in vitro models. And we've been doing a lot of work with three-dimensional models, and this particular one is where we grow the cells in a suspension. So these are low adherent plates. So the cells, in order to survive, attach to each other, and they form these tightly packed, what we call tumour spheres. And you can actually grow those to different sizes and actually look at them. And once they get to about 200, 300 micrometers in diameter, you start getting some hypoxia forming, and as it gets bigger, you get more hypoxic cores, and I'll show you some of that. You can also, this is just, if you were just taking a single cell line, I just call those 3D spheroids. You can also do more complex 3D organoids as well, where you can take tumour material from an animal or even a patient, And in that environment, you've got a much more complex mix. You're going to have endothelial cells in there, you're going to have some immune cells, you're going to have some stromal cells as well as the tumour cells, and probably some stem cells as well. So they're much more complex, but a lot less defined. So we don't use those routinely. It's only sort of at the late stage of development that we'd go down that model. The 3D spheroids are these tightly packed spheroids you can see here. You can also use these spheroids to look at how your particles are affecting cell migration. So you can actually grow them and then gently collect them and place them on plastic and look at the outgrowth, which is what you're seeing here on the outside of these tumour cells. And if you want to see if your nanoparticles interfering with the viability there of the particle versus when they're out in single suspension, you can look at that, or if you're trying to block migration. Another model we use is this 3D spheroid matrix, and this is where we can take the 3D spheroids and actually embed them in a three-dimensional matrix. So why do we do that? We do that because we want to actually study how the tumour, that spheroid in an environment, because if you think in the body, many times these tumours are growing within organs, so they're surrounded by other cells or stroma or a matrix around them as well. So it helps us study those, and we can look at the effects. So depending on the material you're using, you can also use these types of models as well. And you can take all of these actually and collect them, you can section them, you can stain them, you can do RNA analysis or protein analysis on them as well. For those of you who've never seen one of these spheroids form, this is a little movie showing you from a single cell suspension. So these are growing in suspension. These are some tumour cells, some time-lapse microscopy showing the tumour spheroids forming. And see how the cells become attracted to each other. You'll even see ones from a distance starting to come in close and start to attach. They form a very tightly packed network of cells. So, and this is the reason we want to use this. Is I mentioned already about being able to mimic necrotic cores. So the 3D spheroids can be grown. This is what the spheroid looks like. You can even see different density of the material there. What you've got on this outer core of the spheroid is proliferating cells. These are highly proliferating cells. And then you go in, you start getting quiescent cells. These cells are not dividing. And in the center, you get these necrotic cores. And you can actually, and studies have been done to show that as you go to the center of the core, that you're losing oxygen in that model, and also you're getting less nutrients and growth factors in the center of that model. So it's a really valuable model to think about using for some of your work. It can also be used, as we've used it, for looking at penetration of your materials into a spheroid to see how far they can enter. So this is a study we did where we're using a dextran doxorubicin conjugate, and this is just doxorubicin alone. This is confocal microscopy and taking sections through that those 3D models, and you can see that the cells just penetrate the outside of the spheroid when you just have doxorubicin alone. And this was only about, we estimated 50 micrometers diameter, which is only about 1 or 2 cell diameter penetration. But when we actually had the dextran on the doxorubicin, we actually got penetration right through the spheroid. Not the same intensity, but you can see there's a lot more penetration there. And we could actually quantitate it here, where you can see it at 4 and 24 hours, much more doxorubicin inside the cell compared to doxorubicin alone. So this can be quite valuable, and to seeing how your materials are able to do that. We still don't totally understand why the dextran dox was doing that, except all we can think of is potentially the glycoproteins on the surface of the cells were somehow helping facilitate the transport. The other thing that's really advancing, and I guess it's worth everyone being aware of this, is imaging technology is moving at an incredible pace. So many of you would have heard about super resolution microscopy. There's also this new type of microscopy called the light sheet microscope, which actually allows you to take images right through a spheroid and get high resolution images. So I'll just show you one of the movies we recently generated from this system. Here it comes. It's just going to, it's a little bit slow because it's a massive file. But what it's doing, this is taking multiple images throughout the section of the 3D spheroid. And we haven't yet used it for nanoparticle delivery, but ultimately that's why we're working this up. But you'll be able to actually see the nanoparticle uptake at different levels of the spheroid and also in different cells. I mean, this is only stained for actin and the nucleus DAPI, but it can be, you can use different membrane markers and other types of markers when you're fixing these spheroids. But the resolution is incredible, and this is fixed images, sorry, fixed spheroid, but you can also do it on live imaging as well. So it's worth keeping in mind some of these technologies which are advancing that can really facilitate your work and give you better insight. Just see if I can stop the movie. Okay. All right, so I'm going to move away from 3D models to some mouse models of cancer, because I thought it would be beneficial to just talk about the pros and cons of the different types of models. Many of you would be familiar with the subcutaneous tumour models. We'll be talking about disseminated disease model, orthotopic, syngeneic, transgenic. I'll just touch on the patient-derived xenografts as well. But really, which model you can end up using, it really comes down to the question you're asking and what you're trying to address in your system. So you really want to have a model that represents, you know, the reason why you're actually doing the work, you know, whether you're just trying to get general targeting of materials into a tumour, or are you trying to target a particular surface receptor on those tumours. It really does influence how it works. So the tumour microenvironment, like I already mentioned, is highly complex, you know, with many different types of cells. You've got your blood vessels, you've got your stem cells, you've got inflammatory cells, you know, as well as your immune cells, your endothelial cells, stroma. So that's the reason we mainly try and use animal models to better recapitulate what's going on, because there's a limitation, as you saw with those in vitro models, about how much you can do to replicate this environment. And this is just an image from a review that was published a few years ago, just talking about some of the challenges of delivering nanoparticles, the issue about macrophages mopping up your nanoparticles, the leakiness of the blood vessels and the ability to get some materials around the place, and also the excretion by the reticuloendothelial system. So there's been a lot of discussion recently in publications about the amount of material that actually gets to these tumour sites, saying that they estimate around 1% of the material that is delivered gets to the tumour site. I mean, ultimately, if you're getting good efficacy, if only 1% gets there, then 1% gets there. But if you're seeing good efficacy, then I think that's great, because that's ultimately what you want to see. But it would be nice to think about ways to enhancing that delivery effect and maximising delivery. So in terms of subcutaneous models, these are usually just grown. The cells are just injected under the skin. The tumours form, and you know it's quite an easy model to monitor because you can just measure it. So it's incredibly easy to establish. You don't have to be a brain surgeon to actually develop these. It's very easy to train people. They generally are fast growing because they're usually, in this case, you know, we often use cell culture, although if you're doing patient material, it will be a lot slower. You can see the tumours and they're incredibly easy to measure manually. So you don't actually need any sophisticated imaging equipment to measure them. The big disadvantages are the vascular structures may not mimic the primary tumour. And we've actually found this with some of our lung cancer models that we use, that the subcutaneous model, the architecture of the tumour, looks very different to what a human tumour looks like. And the other thing is that the blood vessels often are quite different that you see in, say, compared to an orthotopic tumour that I'll be talking about. So it may not necessarily reflect those tumours, their delivery to a primary tumour. And it also lacks a true tumour microenvironment and architecture. So what I'm talking about is subcutaneous tumour model where we're xenografting. And I'll talk more about models where, you're doing syngeneic models. But in this case, if you're using a xenograft model, this is one of the limitations because the mice are immunosuppressed that you're using, so you're not going to have that intact, complete environment. And then we also use a model called the disseminated tumour model, and this is where we can actually just inject the tumour cells in the tail vein, and they circulate and they form tumours quite rapidly. In this particular model, I'm showing you, this is a neuroblastoma model. It's very fast growing. It allows delivery of the nanoparticles to disseminated disease, so you're not just so you'll have some small nodules, you'll have some larger nodules. We usually use luciferase expressing cells for this model, so we can track roughly where they are. And you find them in a range of organs, and the tumours will vary in size. So again, if you're trying to use particles in delivery to target different sized tumours, it's not a bad, quick model. It's not a true model of metastasis, by the way, but it does give you a sense of disseminated disease. The disadvantages, I guess, it does require some technical skill for the intravenous injections. There's imaging costs, like whether you use a xenogen for luciferase or CT or MRI imaging. It adds costs. And like I said, not a true metastasis model if you really want to be targeting metastatic spread of disease. So here's an example where we used this recently. We had a very specific question we were asking, where we're actually looking at the ability of the tumour cells to be able to cross the endothelial layer. And so we did this tail vein model, and you can see here that in these control samples, there's massive infiltration in the liver. We saw similar results for the lung and the kidney as well. And then when we knocked down our gene of interest, we got massive reduction, even in this tail vein model, of actually the tumours forming. And we could quantitate the luminescence activity, and you can actually identify the tumours inside the liver of the mice. You can actually count those as well. And you can see there's a significant reduction in these tumours. So it's actually a good model if you really have very specific questions you're asking for that purpose. Now, the orthotopic tumour models, and we've moved more and more towards these models for our work. This is where you actually grow the tumour cells in their, the environment where they would have been derived from. So if you're looking at lung cancer, you're growing them in the lung. If you're looking at neuroblastoma, the most common site for that is above the adrenal cortex. So we inject the cells there. For the pancreatic cancer, we inject the cells in the pancreas. So it mainly mimics the environment. The tumour architecture, from what we can see, is similar to the human disease. And it's also useful for studying metastasis. So all the models we have are also metastatic. The big disadvantage is it does take a bit of skill to do the surgery. It's a bit more complicated than just doing a subcutaneous model. You do need imaging tools, like I said earlier, and because you can't see the tumour, you do have to monitor the animals carefully. And the xenograft studies do require immunocompromised mice, so it's hard to determine the immune effects from that model. But, generally, one model will not address all the questions you're trying to you ask. I mean, you can go towards a humanised mouse model. I don't know if some of you have heard about this, where you can constitute the blood of the mice with human cells to make it humanised. But it's an incredibly difficult and time-consuming model to do. So in terms of orthotopic models we've established, this is the lung cancer model, where we inject the cells the other part of the lung. And you can see here, this is normal lung, this pink, and the purple is the tumour. And this is just one of our cancer markers in brown here that you can see. But when you look at the human tumours, the architecture of these tumours is very similar to what you see in a human non-small cell lung cancer. While when we take similar images with the subcutaneous model, these same cells, the architecture is completely different. The blood vessels are completely different. So if you want to mimic the sort of environment, particularly in terms of the architecture and the blood supply, you're better off using these type of models. And this is just going into high zoom here. The other model I mentioned is this neuroblastoma model, where we inject the cells above the kidney. This is also a metastatic model of neuroblastoma. So neuroblastoma is this very aggressive childhood cancer. So 70% of children diagnosed with this disease have already got metastasis. So it's already spread. So survival rates are between 40 and 50% for this cancer. And what you can see here is, the primary tumour is glowing because it's very large. When we take the primary tumour out and we look at the carcass, you can see that you get metastasis to different organs. And we do also know that this model metastasizes to lung, and that's what you can see here. This is staining of the tumour cells in brown here, and surrounded here by some normal lung. So you can actually detect these lung nodules. So again, a really good model if you want to look at these type of things and you want to look at metastasis as well. And this is also an orthotopic model we've recently published on in pancreatic cancer. Pancreatic cancer, for those who don't know, is apart from being a virtually lethal cancer, 5% survival rates, 5-year survival rates for this cancer, it's really rich in stroma. So there's only a few cancer cells in the mix, so they're very stromal rich. So even getting nanoparticles to penetrate that sort of environment can be a challenge. And this is a very nice orthotopic model. It's been validated, and this is where we used some star PEG particles that delivering siRNA, just looking at biodistribution. You can see the heart and lungs, 1 hour, liver, spleen, and the kidneys. And also the liver still retained at 4 hours. But you can see by 24 hours, most of the organs had cleared out the signal, but the pancreatic tumour was still retaining a lot of the signal. So this was quite exciting for us because, like I said, this is a very stromal-rich cancer. You can also, what we did is we also took these tumours and we did frozen sectioning. And what you can see here, this is just CD31, which is the blood vessel there. The blue is the DAPI, and the red is the fluorescent siRNA, showing that we got uptake in the tumours themselves. So that was quite exciting. But, using a mode of modalities is really important for this type of work. Now, the syngeneic model. So the syngeneic model is when you put like with like. So if you're going to do rat on, be rat on rat or mouse on mouse. So here is where we use, say, for example, we've got some non-small cell lung cancer, which are mouse non-small cell lung cancer, and we can put them in a mouse model. And in that model, the nice thing is you can mimic the tumour environment, the tumour architecture, again, very similar to what we see in human disease. And importantly, it's got an intact immune system. So if you want to try and understand how your particles are being influenced by having the immune system in there, this is a nice model to use. But, I guess one of the big disadvantages is that the mouse cells may not reflect the surface markers found on the human cells you're actually really after. So if you've got targeted nanoparticles, that can be an issue. If they're not targeted and you're just doing passive delivery, then that may not be an issue. So, and you're limited to the use of available mouse cell lines, and there's not as many good mouse tumour models as there are human tumour models. And this is the, sorry about, I know you've all just had lunch, but this is the transgenic tumour model. So this is a model that's got a transgene in that causes neuroblastoma. So the 100% of the plus mice develop neuroblastoma tumours. The nice thing about this model is that it actually, it actually forms tumours at the site, but it's above the adrenal cortex, where the majority of tumours form, and it actually spreads in the body as well of the mice. So the nice thing is there's no xenografting required. The tumours originate in their biological environment, and they've got an intact immune system. The big disadvantage is that you normally, to feel these tumours, you've got to have someone who's very well trained in palpating these tumours, and you also need specialised imaging tools to detect these, because we don't have any labels on these tumours. they don't always mimic the human model of disease, but I think for purpose of nanoparticle delivery that it still can be quite useful. And there's enormously high breeding costs because only 25% of mice are ones you can use for this, because the remainder won't have the genotype that you need. So that's one of the disadvantages. But again, it can be very valuable. And then the last model I'm going to mention is the patient-derived tumour models. There's enormous interest for clinical testing and personalised or precision medicine to be testing patient-derived samples. The key benefit is the patient, it's patient tumour material, so it's material that's come straight from the patients that gets xenografted, and it is xenografted. So again, you're using an immunosuppressed animal. The growth rates of the gene expression are usually representative of what you see in the primary tumour or the original tumour. But there's also disadvantages, like I said, immunosuppressed mice. The tumours often grow very slowly. So we're all very used to tissue culture where cells proliferate because they've been going for years and years and years, and 24 hours, 36 hours, the cells have doubled. Well, normally in a human body, cells don't multiply that fast. And so when you put them into these models, they do grow much more slowly. So they can actually take months and months. Not all the patient samples also will engraft. But once you've got them established, once you know they're working, then re-engrafting them usually works. You've just got to check that they're not losing their phenotype. It's incredibly time-consuming and expensive. But again, if you have a key question to ask, then it's worth considering this type of model. So what I was trying to get, I guess, across today is that, cancer delivery, for nanoparticles, this one size is not going to fit all. The biology of different types of tumours varies enormously. And I guess translating nanomedicine to the clinic, we really need to understand the tumour type and pathophysiology of these tumours. We need, I guess, ultimately a patient-focused design of nanomedicines, I guess akin to some of the targeted type therapies. And I think we also need improved testing models to bridge the gap, because we know that there's a big dropout rate once these things get to clinical trials. And if we have good models and we've got good systems in place to be able to do the preclinical testing to minimise things that shouldn't be getting to the clinic, that would be great. And it could be that just some select patients are likely to benefit from these treatments, and you may not hit everyone. So here's my little cartoon. Basically, I guess we're trying to deliver these materials. In our case, we've suppressed genes that are involved in drug resistance to sensitise them to chemotherapy to ultimately eliminate the tumours. And so let's hope between all the brains in this room and elsewhere that one day we're able to achieve this, because there's many cancers that are desperate for some better solutions than what we've got now. So thank you for your time.
Thank you very much for the explanation of growing costs in the different